may we ask Rik to come on stage, please?
So the next speaker is Rik Lindeboom. He's joining us from the Netherlands Cancer Institute. He's actually trained over here in Nijmegen at the Radboud, spent some time at the Wellcome Sanger Institute in Cambridge, and now at the NKI. And he's applying quantitative proteomics and genomics to understand the interaction between tumors and their immune environment. So thank you.
That's right. Thank you very much. Thanks very much for the kind introduction. Let's see if I do get this to work. That's the this click forward. Back, yeah. Yes, thank you very much. Thanks so much for the kind introduction and for the invitation for this nice platform. I'm a junior group leader at the NKI now for 2 years, and today I will share some of the work that my lab has been working on for the past 2 years. So, in general, we are interested in a very fundamental question in life, which is how it is possible that the same protein can have different functions at different concentrations. And this is, of course, a very basic question because proteins, they are sort of involved in every possible biological process that you can think of. So within my group, we try to focus this question towards 2 different scientific domains, one being transcription factor binding and the other one being understanding how antigen abundances on the cell surface are regulated and how that relates to T-cell binding. And the reason that we focus on these 2 questions is because they are beautiful examples of where the same protein or the same peptide can have different consequences for a cell depending on the concentration. And I will sort of explain that more in detail later in my talk. But as you all know, when you're setting up your lab and you have all these funky questions that you would like to answer, the first thing that you need to do is you actually need to set up the technologies that allow you to study the questions that you're interested in. So for this talk, I will mainly focus on that. So starting with transcription factors. So transcription factors are, of course, proteins that bind to the DNA to regulate gene expression. And the way that this typically works is that upon a stimulus or a perturbation, the amount of transcription factor in a cell, it goes up in concentration, and that then allows the transcription factor to bind to certain DNA loci, which activate specific gene signatures. But really understanding how this change in concentration leads to a change in binding is something that is quite hard to study. And because of this, we can't really answer very simple questions, which, for example, I wrote down here, how is it possible that a healthy cell is happy and about having 10,000 c-MYC molecules in the nucleus? But then if you go to 100,000 c-MYC molecules, you all of a sudden become a cancer cell. The reason that we're limited in studying this is because there's limited methodologies out there that enable us to answer this dosage-dependent transcription factor binding question. And the key thing that we needed to solve to sort of address this properly was that the current methods, they all work with naked DNA, but of course transcription factors, they function in the context of chromatin, which is crucial for guiding transcription factor binding. So we had to come up with a methodology that allowed us to sort of study transcription factor binding over concentration in the context of chromatinized DNA. And the way that we approached this problem was conceptually relatively simple, where we took nuclei, which of course contain chromatinized DNA, and then we permeabilize them, and then we incubate those permeabilized nuclei with a titration series of transcription factors of interest. These transcription factors, they have an affinity purification tag, which then allows us to do a ChIP-seq experiment to measure the binding profile at every concentration that we expose these nuclei to. And then this is sort of what the data looks like, where you can then identify at which concentration of the transcription factor binding occurs at every locus in the chromatinized genome. So this is what the actual data looks like. Here showing for the BRD2 locus, where you can see that between 125 and 250 nanomolars of YY1 binding occurs at this locus, which indicates that the binding affinity to this specific gene is around 150 nanomolars. But of course, the real cool thing about this method is now that we can study these differences in binding in the context of the epigenome. And we can study how the epigenome is regulating dosage-dependent transcription factor binding. And this is a very busy slide for which I apologize, but this is sort of the analysis that we did here. Whoops, where we profiled 4 different transcription factors, so 4 different heat maps. And the first thing you can see the quantified binding affinity over all these different genomic loci. So every row in these heat maps is a binding site in the genome. And the first thing that we looked at is the annotated regulatory features that each binding event was happening in. And this is denoted here in the color code to the left of each heat map. And you can see by what is quite obvious is that dark green is overrepresented, and this means the promoter regions. So at least for these 3 transcription factors, we see that nanomolar binding affinities happen nearly exclusively at promoter regions. And then when we look into these regulatory sites, what epigenetic features are explaining the nanomolar binding affinities, we can see that active chromatin regions, but in particular DNA accessibility, is really a facilitator of nanomolar binding affinities. The obvious exception to this story is FOXA1. Now, FOXA1 was also the only pioneering transcription factor that we tested, and we think that this pioneering capability, where it is able to bind to the DNA and facilitate its own opening up of the chromatin, is actually explaining this difference here. The importance of the chromatin becomes even more important when you start looking at a dynamic cell system, where we've here differentiated embryonic stem cells on the left to neuroprogenitor cells to the right. And then here you can see the binding at these different concentrations of, I think this was SP1, and where you can see that we're able to detect cell type specific binding affinities. And this is, of course, the identical DNA content is within these cells. The only difference is the cell state, and with that, the epigenome. And indeed, if we then look what is explaining these cell type specific nanomolar binding affinities, we can see that DNA accessibility, pre-existing DNA accessibility, it can explain this. But then to come back to sort of my original question, how is it possible that a transcription factor can lead to different phenotypes depending on its concentration? We try to couple back binding to gene regulation. And the way that we try to analyze this is by doing a gene set enrichment analysis to look for gene sets and biological processes that are specifically bound at a certain concentration of, I think, in this case, SP1 again. And an example of such an analysis you can see here, where on the top you have gene sets that are bound at low concentrations of SP1, so high affinity binding sites, and at the bottom you have gene sets that require much higher concentrations of SP1. So really indicating that by changing the concentration of SP1, a cell can activate different gene sets. And that brings me to the conclusions of the first part of my talk, where I showed you BANG-seq, which stands for binding affinities in native chromatin by sequencing, by the way. Which is a method to measure absolute binding affinities for transcription factors in chromatinized genomes. We've showed that DNA accessibility is a major regulator of affinity modulation, and these differences in affinities across the genome allow cells to exert concentration-dependent changes to gene activation. Now this was work that I started when I was still in the lab of Michiel Vermeulen, indeed, and then I continued together with Hanna Nijkens to work on this throughout my postdoc and into my new lab, where we're now taking this technology to expand on it further, where we're studying oncogenic and T-cell transcription factors. We're also trying to expand the amount of measurable binding parameters to also study association and dissociation rates, but also binding energies that are required, with the real goal of understanding how each chromatin feature links to certain changes in binding kinetics. And then switching gears a bit, as I said, I was also going to talk about antigens, which are again peptides that are exerting this concentration-dependent phenotypes. Antigens are, of course, presented on the cell surface of a cell, and they enable the recognition by, specific recognition by T cells. But for this recognition to happen, the concentration, it needs to be spot on. So it can be too low, of course, because then you don't get recognition at all. It can also be it can't be too high either, because that will lead to a dysfunctional T cell response. But really understanding what is regulating this eventual concentration for each of the antigens that can be presented by a tumor cell, something that we're not really able to. And one of the reasons for that is that there is actually a very poor correlation between protein expression and antigen abundances. This is something that my lab is very interested in studying, and the way that we try to understand this is by measuring proteome kinetics and degradation kinetics and trying to link that back to the abundances of different antigens. And this is on the proteomic side relatively easy to do because there's many tools to do this. But on the immunopeptidomic side, so on the antigen repertoire side, this is much harder. And the main reason for that is that the current state of the art in immunopeptidomics, it uses antibodies and a lot of cells, so a lot of antibody and a lot of cells to pull down the MHC class 1 molecule, elute off the antigens to then sequence what is loaded there. That leads to very good data, but it is poorly scalable because of the sort of practical limitations of these requirements. So our goal is really to measure a lot of variation happening in the antigen repertoire side, and this is really now refraining us from doing this because this methodology is not scalable enough. So we set out to sort of look for alternatives, and we actually ended up with a much older method, but that it's not so often used anymore, which is called mild acid elution. And this method makes use of the acid label characteristics of the B2M subunit of MHC class 1, which falls off if you treat the cells with a pH of 3.3. And with that, the conformation of the HLA molecule, it changes, which elutes off the antigen. Now, and because this is sort of a single-step reaction, we thought, well, if we now put a resin that captures these peptides and that is mass spec compatible, right after this step, would this then be suitable for a very low input and minimal loss workflow to profile the immunopeptidome? So this is sort of a schematic of the workflow that we ended up developing. So it's a one-pot reaction where we only do the elution of the antigens of the MHC class 1 molecules into the supernatant, and then we directly transfer that in a single pipetting step onto a filter that is directly above a C18 resin that captures antigens that captures peptides for mass spectrometry analysis. If we then look what these antigens look like, what the peptides look like that have eluted off the cells, you see this very typical amino acid sequence where the last and the second residue are sort of predefined, and this is very characteristic of an immunopeptidome because these are the anchor residues of immunopeptides. What is important is that if we do this in cells that cannot present antigens, we don't see this pattern at all. What is also nice to mention is that the cells actually remain intact on top of this filter, and that actually allows us now to take the cells and actually process them for whole proteome profiling, really making this a multi-omics workflow where you get joint proteome and immunopeptidome profiling of the same small sample. And this is just a proof of concept where we tested this concept. We've treated cells with TGF-beta, and then here you can see the changes on the proteome level where we can still measure up to 10,000 proteins. Here we see the quantitative changes in the immunopeptidome where we have good coverage as well. And then this is an integrated analysis where on the x-axis you see the changes on the proteomic level, and on the y-axis you see the changes on the immunopeptidomic level, where you can again see that there's poor correlation here, really underscoring the importance of doing these measurements. Now, of course, this was TGF-beta stimulation in a single setting. That's not really the goal of this method. As I said, we really want to do this at a large scale to really capture substantial immunopeptidomic variation to then link that back to what we see in the proteome. And one of the things that we aim to do is to do this in large-scale clinical cohorts of tumor samples. But we actually think that our method is so scalable that we cannot just look across many different patients, but even within a single tumor, look at intratumoral heterogeneity by chopping up the single tumor. In this case, it was a kidney cancer tumor, into tiny fragments of a millimeter cubed, and then measuring the immunopeptidome and proteome of every of these fragments to look whether the antigen repertoire is changing across this single tumor. And sort of the first, I'm showing you one of the first results from this, where you can nicely see the dispersion, so the variation of the proteome across this tumor. But now we can also capture this on the immunopeptidomic level. And this is still work in progress. We're still trying to figure out how to handle missing values here, which is why there's not so many dots here. But this will, I'm sure we'll figure this out. And this, I think, is really underscores the power of this method now. So summing everything up, I hope I showed you that mild acid dilution-based immunopeptidomics is a potentially very low input and scalable method to do cost-effective immunopeptidomics. We come in at a bit less than €5 per sample now, which is really orders of magnitudes less than the antibody-based method. And this, we think, now really enables large-scale immunopeptidome profiling in conjunction with proteome profiling to understand what is regulating antigen presentation. And this is exactly what we aim to do. So in large in vitro perturbation screens, as well as patient cohorts, we will try to profile this at scale to model these changes. Something that I didn't have time to talk about yet, but we've also developed a proximity labeling assay to measure the proteins involved in the T-cell antigen, in the T-cell tumor cell interaction. Where we're now mainly looking at the proteins that are involved in this interaction, but we think that this method is actually so low input compatible that we can link this proximity labeling with immunopeptidomics to measure the tumor antigens that are involved in the interaction between tumor infiltrating lymphocytes and tumor cells. So that's also an outlook of this method. And with that, I would like to thank the people from my lab, in particular Mayuka, who set up the immunopeptidomics workflow. As I said, the BankSeq was developed together with Michiel and Hanna, and now also Sweta is continuing this work in my own lab. Thanks to my collaborators, these are my funders, and thank you for listening. I'm happy to take any questions that you might still have.
Thank you very much, Rik. Questions, indeed.
For your first part, did you get to see in the levels of concentrations differences between pioneering transcription factors and other transcription factors that are not fully necessary on accessibility, for example, these sort of differences?
Yeah. So that was very obvious when we started looking at FOXA1, which was the only pioneering transcription factor that we considered for the initial study. This is something that we're planning to look into deeper because, of course, N is 1 right now. But it seems that for FOXA1, DNA sequence is really key in determining dosage-dependent binding, whereas for all the other transcription factors we've seen, the epigenome is the main sort of regulator of binding, and then DNA sequence is actually secondary to that. So yes, pioneering versus non-pioneering seems to be a very big difference.
All right.
Yeah.
Okay, so maybe in the interest of time, thank you very much.